Good evening. I'm Matt Rudolpher, Chair of the Washington Psychiatric Society's Continuing Medical Education Committee, and I'd like to welcome you to the Society's monthly Wednesday evening webinar series for November. In the excitement that greets the approval of each new medication, it's easy to overlook the many months and years of hard work and resources with careful regulatory oversight, often unseen, that went into reaching the point of marketing. And with a current pipeline bulging with potential new meds, with a dizzying array of novel mechanisms of action and challenging mixes of potential benefits and risks, the role of the Food and Drug Administration in ensuring that the medications we prescribe for our patients are safe and effective is more critical than ever. To take us inside the FDA, the WPS is delighted and honored to welcome this evening Dr. Tiffany Farchione, Director of the Division of Psychiatry, Office of Neuroscience in the FDA's Center for Drug Evaluation and Research. Dr. Farchione received her medical degree from Wayne State University in Detroit and completed adult psychiatry residency and child and adolescent fellowship training in the University of Pittsburgh's Western Psychiatric Institute and Clinic, later serving on the faculty at Pittsburgh. She is board certified in both general and child and adolescent psychiatry. Dr. Farchione joined the FDA in 2010 and for the past 4 years has served as director of the Division of Psychiatry, providing oversight of new drug review for all psychiatric drug development activities conducted under investigational new drug applications and the review of all new drug applications and supplements for new psychiatric drug claims. Her presentation this evening is entitled Inside the FDA, A Clinician's Guide to the Drug Approval Process. Let me just note, as Dr. Farchione gives her presentation, if you have questions, please enter them into the chat, and we'll save them for the end. Take it away, Tiffany.
All right. Thank you. Thank you so much. All right, let me just share my screen. All right, and if somebody can confirm that you can see the slides. All right, great. Okay, so as Dr. Rudolpher mentioned, my name is Tiffany Farchione, and I'm the director of the Division of Psychiatry at the U.S. Food and Drug Administration. And I'm here to talk to you tonight a little bit about what exactly the FDA actually does and how we do it. So, everybody seems to know that the FDA is the government agency in charge of approving new drugs. But most folks outside of the world of drug development have very little understanding of what's actually involved in that process. So tonight, my job is going to be to shed some light on that. Right. And to start, obviously I have the standard FDA disclaimer, and like most feds, I have no financial interest to disclose. All right, so tonight I'm going to start with a really quick introduction into who I am and how I got to the FDA. Then I'll talk briefly about how FDA came to be and some of the legislation behind our main regulatory authorities and an overview of how we're organized. After that, I'll talk a little bit about our oversight of clinical studies during drug development and how we evaluate clinical study data to determine whether the benefits of a drug outweigh its risks. And then I'll wrap up by talking about where you can find FDA resources and how you can be part of the process. All right, so I figure I should start with my own introduction because, the chances are that you've probably never met someone who works at the FDA before, although I do see somebody on the line who maybe knows a little bit about this. But in any case, we are in the DMV, so your chances are better than most other audiences for this, but still, it's not exactly one of the well-defined career paths that's presented to you in med school or in residency. And I know that I certainly never thought about it. So in fact, I had a plan. And to borrow a metaphor from my old department chair, David Kupfer, I knew what my t-shirt was going to say. So the idea of the t-shirt metaphor is that you should be able to describe yourself or your research interest succinctly enough that it could fit on the front of a t-shirt. And mine was going to say that I was a physician scientist with expertise in pediatric mood disorders and functional neuroimaging. And I was totally 100% absolutely sure about this. I had no doubts whatsoever. So I started on the usual path and I picked a residency program with a strong research track. I trained in Pittsburgh. You can't get stronger than that. Not that I'm biased. I identified actually a team of mentors, and I made sure that I had protected time for my research. After residency, I entered a T32 funded postdoc program. I applied for foundation funding and got it. I started collecting pilot data for my K. And then, the next steps were that I was going to apply for my K award, I was going to apply for an R01, and then I was going to be a principal investigator. And, like I said, this is the usual path, and it may be well established, but it's one heck of an uphill slog. And to be honest, while I was doing that, I was miserable. So fortunately for me, I happened to run into an old friend from medical school at the ACAP conference one year, and he had just started a job at FDA at the time. And he told me that he thought that I would really love it there. And I kind of blew him off, because I was like, no, I know what I'm going to do with my life. But when I decided that I didn't want to write my K, I remembered the conversation and I gave him a call. And then shortly after that, I interviewed, and the rest, as they say, is history. All right, so that's my history. So I want to shift gears now to the agency's history and start by going back to the very beginning. So although FDA is currently part of the Department of Health and Human Services, we started out as the Division of Chemistry within the Department of Agriculture. The Division of Chemistry began investigating the adulteration of agricultural commodities as early as 1867. And around that same time, you had your muckraking journalists who were exposing the hazards of the marketplace, both in terms of adulterated food and quack medications. Things came to a head with the publication of Upton Sinclair's The Jungle in 1906 that documented the horrors of the meatpacking industry. And after that, that led Congress to take action with the Pure Food and Drug Act. Now, the act prohibited the interstate transport of unlawful food and drugs under penalty of seizure of the questionable products and or prosecution of responsible parties. So now, instead of being just the Department of Chemistry, the Division of Chemistry, we were now the Bureau of Chemistry, and the Bureau was in charge of administering the act. Under the act, drugs were now defined in accordance with standards of strength, quality, and purity in the U.S. Pharmacopoeia and the National Formulary, and they couldn't be sold in any other condition unless the specific variations from the standards were plainly stated on the label. And foods were not defined according to analogous standards, but the law prohibited the addition of any ingredient that would substitute for the food, conceal damage, or pose a health hazard, or constitute a filthy or decomposed substance. Okay, that's the remote history. So even though the Pure Food and Drug Act required that drugs meet certain standards, those standards had nothing to do with safety or efficacy. So there were still a lot of worthless cures floating around out there. And it took a massive tragedy in order to change that. So in 1937, the S.E. Massengill Company began marketing elixir sulfanilamide. And this was an oral liquid antibiotic that was created using diethylene glycol as a solvent. Unsurprisingly, it ended up killing people. It killed about 100 people, including many children. So Congress responded to public outrage by passing the 1938 Food, Drug, and Cosmetic Act, or FD&C, which required companies to perform animal safety tests on their proposed new drugs and submit data to the FDA, we now were FDA, before being allowed to market their product. So now, based on this authority, FDA is able to regulate drug safety. So that is the first legislation that really brought about some of our core regulatory responsibilities. But it took another tragedy to give us the authority to evaluate efficacy as part of the drug approval process. So thalidomide was marketed in Europe and Canada as a treatment for anxiety, trouble sleeping, tension, and morning sickness. But in the United States, it was a junior reviewer at the FDA in one of her first assignments who blocked its approval. So Frances Kelsey was concerned about an English study that linked thalidomide to peripheral neuritis, and she kept asking the manufacturer to explain the findings. She also asked them for data showing that the drug would not be harmful to the fetus. So remember, at this point, safety is our only leverage. And she used that leverage to effectively block the approval of thalidomide in the United States. And in the meantime, the drug began to be linked with birth defects, and it was eventually pulled from all markets. So the thalidomide tragedy prompted tougher rules for testing and licensing of drugs. And here in the US, those rules were codified in the Kefauver-Harris Amendment to the FD&C. And these new regulations strengthened FDA, among other ways, by requiring applicants to prove efficacy and to disclose all the side effects encountered in the testing. So the standard outlined in the amendment required manufacturers to establish a drug's effectiveness by substantial evidence. And substantial evidence was defined as evidence consisting of adequate and well-controlled investigations, plural. And we'll come back to this a little bit later. But as a result of all of this work, Frances Kelsey was awarded the President's Award for Distinguished Federal Civilian Service. And ever since then, FDA has been responsible for evaluating both safety and efficacy of drugs. All right. So fast forward a few decades and lots of additional legislation, and this is what FDA looks like today. So now we have authority over not just food and drugs, but also biologics, medical devices, tobacco, veterinary medicine. But today I'm just going to focus on drugs and what we do in the Center for Drug Evaluation and Research, or CDER for short. So CDER is organized into several offices, and we're in the Office of New Drugs, but there are also offices overseeing generic drugs, pharmaceutical quality, surveillance and epidemiology, and more. Office of New Drugs is then further divided into smaller offices based loosely on body system. So my group is part of the Office of Neuroscience, which includes the Division of Anesthesiology, Addiction, and Pain Medicine, 2 neurology divisions, and the Division of Psychiatry, or DP for short. So DP is responsible for all of psychiatry. So this includes the obvious indications like schizophrenia, depression, bipolar disorder, but we also review applications for the behavioral manifestations of certain diseases. So, for example, treatments of Alzheimer's disease are reviewed by the Division of Neurology 1, but we get the behavioral and psychological symptoms of dementia. So, your things like agitation, aggression, psychosis. All right. So I'm going to delve into the nitty-gritty of the drug review process in a minute. But before I talk about what we do at FDA, I want to talk a little bit about what we don't do. Because there's a lot of misconceptions out there. And to illustrate this, I'm going to quickly walk through a relatively recent example from one of the final episodes of the TV show New Amsterdam. So bear with me for a moment here. But, I think as physicians, we all know that medical dramas can strain credulity. I can still remember watching one of the first episodes of Grey's Anatomy when, this world-renowned cardiothoracic surgeon scrubbed in on an appendectomy, which, obviously is ridiculous. But no more ridiculous, I think, than when the surgery interns would show up at the hospital in broad daylight. When you talk about willful suspension of disbelief, that can only really go so far. But for me, the storyline in this episode was particularly cringeworthy. So it kicks off with Dr. Max Goodwin announcing New Amsterdam's partnership with a pharmaceutical company and the results of their clinical study that found a cure for bulky large cell lymphoma. But we need some drama, so enter Dr. Elizabeth Wilder, Max's colleague and current love interest. So Max can tell that something is wrong, and Wilder tells him that the trial is a sham. And she goes on to explain that even though the drug cured 89% of the patients in the trial, the study population was all ages 35 to 65 and 89% Caucasian. And she's worried that without testing on a more diverse population, they can't be sure that it's safe for folks who aren't white. So she doesn't want this to be the next thalidomide. And Max has to explain to Wilder's ASL interpreter, and conveniently to the audience, that thalidomide is a drug from the 1960s that they gave to pregnant women for nausea, but we didn't know it would cause birth defects because no one tested it in pregnant women. And they decided that they can't release the data from the trial and that they're going to kill the cure for cancer. So Max and Wilder confront the perennial medical drama bad guys, Big Pharma and the hospital administrator. So Pharma guy says that demographics don't matter because the drug targets a specific genetic defect, and the administrator is incredulous. She says that it may not have been tested on all of mankind, but there's irrefutable proof that it saves lives. Some lives. So Max responds, I don't want to own a trial that says whites only. So now comes the big twist, right? Pharma guy says, but these were your parameters. Wait, what? So Max and Wilder figure out that Dr. Sharp, Max's previous love interest, set the trial parameters so that Max would be part of the study a few seasons ago when he had cancer. So they hatch a plan. They tell Pharma guy that they're going to submit the data, but they're also going to immediately start a new trial enrolling more diverse participants. And they acknowledge that the inequities are on them, but they tell Pharma guy that his company is going to profit, so they will be funding the study. So obviously, Pharma guy balks. He says, we bring drugs to market to make money, not to hemorrhage it. And Max says, well, if you don't fund it, we bury the results and no one gets the drugs. So Pharma guy relents. They have a deal. And as a bonus, the whole experience reinforces for Max and Wilder that they're perfect for each other. So everybody lives happily ever after. Except for me, because I'm at home yelling at the TV because of how inaccurate the whole storyline was from start to finish. And it's hard to know where to begin. But, just for a few ideas here. It's drug companies, not the individual study sites, that set the inclusion and exclusion criteria for trials. We review the protocol before the study is even started. So if we saw inappropriately narrow inclusion criterion, we would tell the company to fix it. So, to improve generalizability of the study results or to provide a reasonable scientific justification why they choose not to fix it. We use animal data to give us information about potential fetal malformations. A better but less dramatic example of the importance of diversity in clinical trials might be something like zolpidem, where the recommended dose for women is half that of the recommended dose for men, and we didn't figure that out until after the drug was on the market because the clinical studies were about 80% male. The drug company owns the data. A study site doesn't get to bury it. And if the drug is improved despite the lack of trial diversity, we can actually require post-marketing studies if we have a safety concern. So that's just the tip of the iceberg. And I could honestly populate a multiverse with alternative storylines addressing the various inaccuracies here. But instead, I'm going to just start by walking you through what we actually do. All right, so this is an overview of the drug development timeline. So companies developing a drug for clinical use start with basic research in animal studies. Once they're ready to start studies in humans in the United States, they submit their investigational new drug application, or IND, to request permission from the FDA to conduct the clinical study. At that point, we then have 30 days to review the protocol and determine whether it's safe to proceed. After that, the company has to send us the protocols for every study before they begin, but they don't have to wait the 30 days for us to review it anymore. They can start the study as soon as they submit the protocol at that point. But if they're going to want that study to serve as one of the adequate and well-controlled investigations necessary to demonstrate efficacy, the chances are that they've already asked our advice about the study design before submitting the protocol, and they're probably going to wait a few weeks to see if we have any additional advice after our review, which we frequently do. So there's a lot of back and forth engagement between companies and the FDA during drug development. Once the clinical studies are done and the company feels like they can demonstrate both safety and efficacy, they submit their new drug application, or the NDA. So the application goes on a clock, and we have to make a decision within 6 to 12 months. The clock depends on the application type and the potential for the drug to address an unmet medical need. So a brand new drug gets a longer clock than a new indication for an old drug, for example, because there's a lot more information we have to review with a brand new drug. But something that can address an unmet medical need is going to get a shorter priority clock because of the urgency of getting that out to address a public health need. So if we determine that the benefits of the drug outweigh its risks, then the medication will get approved. All right, so that was the 30,000-foot view. And now I want to take a closer look. And I think for that, it's good to start with the review team. So every application is reviewed by a large multidisciplinary team. We have a regulatory project manager who coordinates the review process and communicates with the sponsor. Our CMC team evaluates the drug substance and its synthesis. They also evaluate dosage form, strength, excipients, impurities, and the stability of the drug over time. Our Pharm Tox team reviews all the preclinical studies, including, for example, the animal toxicology studies. They let us know the target organs for toxicity and the safety margins to the proposed human dose. The clinical pharmacology team evaluates the drug's absorption, distribution, metabolism, and excretion. And they also evaluate drug-drug interaction studies and studies evaluating the impacts of organ impairment on PK parameters. The clinical reviewer evaluates the study protocol itself. That includes everything from the inclusion and exclusion criteria to the proposed dosage and the duration of treatment, the safety monitoring, the endpoints. The stats team will get involved at the IND stage if it appears that a study is intended to serve as an adequate and well-controlled study. They need to evaluate the statistical analysis plan early on and ensure that it's appropriate. Sometimes we have consultants involved for specific issues, like we get our clinical outcome assessments folks involved for review of novel study endpoints, or our controlled substances staff will weigh in on abuse liability. And then there's me. I provide oversight of the entire process and try to make sure that we're giving similar advice across similar programs, help the team think through difficult questions or reach consensus when there's a difference of opinion, and so on. And we do this for every clinical protocol throughout the drug development process until finally the studies are completed and the company files their new drug application. And then the whole team gets together again and reviews all of the results. But we'll get there. So I also want to talk a little bit about what a typical day looks like for an FDA reviewer. It's a desk job. We're still mostly working at home. We go in about once a week now, although I think that's probably going to change pretty soon. The work itself involves lots of reading, lots of writing, lots of thinking. We have lots of meetings, but I almost never think that a meeting could have been an email. When the review team gets together for a meeting, it's generally productive and collaborative. We do have congressionally mandated deadlines for most of the work that we do, so that can be a little bit stressful at times. But there's always something new, so that part makes it super exciting. And basically, I describe it as all the fun parts about research, but without having to write any grants, and you don't have to run any of the studies yourself. So, for me, that's the dream job. But getting back to the work itself, what do we do with all of that data? So when reviewing new drug applications, we consider the treatment indication, the available treatment options, and evidence for both the effectiveness and safety of the proposed new drug. And if the drug is assessed to be effective, and if the safety concerns can be adequately managed, then we consider that to be a favorable benefit-risk balance that supports marketing approval. All right, so I've mentioned this a couple of times now, but to support a marketing application, a company has to provide what we call substantial evidence of effectiveness for the drug. And that's the standard that I mentioned earlier, the one that was established by the Kefauver-Harris amendments, which said that drug manufacturers are required to establish effectiveness by substantial evidence, and that substantial evidence is defined as evidence from adequate and well-controlled investigations. So because of that plural, the standard has been interpreted to mean at least 2 positive, adequate, and well-controlled studies. But there's some flexibility in that standard. So for instance, it's going to be really difficult to conduct 2 large, adequate, and well-controlled studies in a rare disease population. And luckily, we have a guidance document that outlines our standards and provides some examples of the types of situations in which we might accept something other than 2 studies. And by the way, at this point, I want to note that we regularly publish guidance documents to help facilitate drug development. The last thing that we want is for drug companies to be flying blind. They need to know what our expectations are so that they can design their trials in a way that will yield interpretable, meaningful, and usable data at the end of the day. So if you've been paying attention, you may have noticed that I've mentioned this concept of adequate and well-controlled a few times. 6 actually at this point, but that's, who's counting. But the features of an adequate and well-controlled study are actually listed in the Code of Federal Regulations. And I'm just going to list a couple of the criteria here. So a study has to use a design that permits a valid comparison with a control to provide a quantitative assessment of the drug effect. So typically, but not always, this means a placebo-controlled trial. Adequate measures are taken to minimize bias on the parts of the subject, observers, and analysts of the data. So, for example, this is where blinding would come into play. And the methods of assessment are well-defined and reliable. So you can't just ask a patient, are you feeling better, and tally up a proportion of yes answers in each group. You have to have a proper assessment of what you're trying to treat. So let's say a company has completed at least 2 adequate and well-controlled trials, they have positive results, and they think the safety profile of the drug is favorable. At that point, they submit their new drug application and we begin our review.
I'm bringing you dinner.
Oh, I'm sorry. If folks can please mute, that would be helpful. So in any case, all of the team members that I mentioned earlier are now involved at this stage. But now, instead of reviewing the protocol, they're reviewing the results of all the studies and coming up with an integrated assessment of the benefits and risks of the drug. And if the benefits outweigh the risks, then we can approve it. And I think the easiest way to talk about what we do when we're reviewing a new drug may be to frame that in the drug label. And so for illustrative purposes, this is the table of contents from one of our recently approved drugs. And if you didn't know, the standard format for drug labels was revised in 2006 with the physician's labeling rule. So at this point, every drug approved since 2006 is in this format, and a good chunk of our older drugs have had their labels converted as well. So if you're looking for indications and usage, that's always right up front in section 1. If you want to know about pediatric use, that's in section 8.4. A description of the clinical studies that supported approval is in section 14, and so on. So going back to the members of the review team and how their work informs the prescribing information. The findings from the CMC reviewer are going to be reflected in sections 3, 11, and 16. That's where you'll find what the drug is, what kind of excipients are in the drug product, what it looks like, how you store it, etc. The non-clinical team evaluates the animal studies that inform parts of section 8, the receptor occupancy studies that inform 12.1, and all of the data that goes into 13. The clinical pharmacology review informs our recommendations for dose adjustment based on intrinsic and extrinsic factors. ClinPharm also evaluates the drug-drug interactions, pharmacodynamics, and pharmacokinetics. And the clinical and statistical teams review the clinical studies together. So not only have we reviewed the studies before they started and provided oversight during the studies, for example, by reviewing adverse event reports, now we get the subject level raw data. We rerun the company's analyses to make sure that we get the same results. We perform our own exploratory and sensitivity analyses. And the company will send us the draft label with their submission, but we usually provide substantial edits to that. So ultimately, we write the description of the trial design, endpoints, and results in section 14. The population in the studies and the assessments are going to inform the indications and usage section. Typically, the dosage and administration instructions will mirror the way the drug was administered in the clinical studies. And finally, we also get the raw data from the adverse event reports. We audit the data to make sure that adverse events are appropriately characterized and grouped for analyses. And we do our own analyses of the data to inform sections 4, 5, and 6 of the label, plus any box warning if necessary. And I really want to drive this point home because what we do is not simply peer review. Did the drug company send us the study reports with all of their analyses? Sure. But do we take that at face value? Absolutely not. We get the raw data and we do our own analyses. We verify, we dig, we look at the application from all angles. And then on top of that, we have another team that reviews the label when we're nearly done with it to look for any language that's promotional, which we then edit further. So nothing makes it into that label unless we've confirmed its accuracy. All right. So sometimes we may approve a drug, but still have some unanswered questions. And we can ask for additional studies. Those are typically negotiated with the company and agreed to in advance. And those are called post-marketing commitments. In certain circumstances, we have the authority to require additional studies. So for example, the Food and Drug Administration Amendment Act, or FDA as we call it, allows us to assess known serious risks related to the use of the drug or signals of serious risk related to the use of the drug, or to identify an unexpected serious risk when available data indicate a potential for serious risk. So basically, we can require post-marketing studies to evaluate a safety signal. We also have the Pediatric Research Equity Act, or PREA, which gives us the authority to require pediatric studies as well. Now, one more circumstance in which we can require studies when a drug is approved is via the accelerated approval pathway. And this pathway allows for earlier approval of drugs that treat serious conditions and fill an unmet medical need. But instead of already having direct clinical evidence of benefit, those approvals are based on a surrogate endpoint. So a surrogate is a marker like a laboratory measurement or radiographic image, physical sign, or other measure that's sought to predict clinical benefit, but is not itself a measure of clinical benefit. And so the use of a surrogate endpoint can considerably shorten the time required prior to receiving FDA approval, but then we need to require those follow-up studies to actually confirm that benefit. But, we don't have any examples of that in psychiatry. And, given our sort of limited knowledge of pathophysiology, I think that we're a long way away from any kind of surrogate endpoint-based approval for a psychiatric indication. All right, so it's important to know that our review doesn't end when the drug is approved. So despite our vigilant pre-market review, active post-marketing surveillance of drug adverse effects is also essential. And we have an entire office dedicated to safety and epidemiology with divisions dedicated to pharmacovigilance. Because we can't possibly identify all the side effects of a drug based on the pre-approval studies that involve only, several hundred to several thousand patients. So we maintain a system of post-marketing surveillance and risk assessment programs to identify adverse events that didn't appear during the drug approval process. And then we use that information to update the drug labeling and on rare occasions to reevaluate the approval or marketing decision itself. So I've given you kind of a whirlwind tour of what we do at FDA. So now I want to point you to some resources where you can, go ahead and find out some more information on your own. People think that the FDA is so mysterious, that we're our own black box in a way, but we're actually incredibly transparent. You just need to know where to look. So to that end, one of the first sites that I want to tell you about is the Drugs at FDA database. And from here, you can look up the label of any approved drug. That in itself might not be super exciting. You're probably thinking that you can just Google the drug name and get the label, but fun fact, the search engine algorithms don't always take you to the most recent version of the label. So if we've added new safety information or approved a new indication, you might not see that if you rely on Google. So here you can go directly to the source. And as a bonus, not only can you find the most recent label, the site also links to older versions of the label, approval letters, and the actual FDA review documents. So, I don't really imagine that most folks want to read, 200 or 300 pages dissecting every aspect of a drug development program. But if you're curious, it's right there. Okay. Sometimes during our review of a new drug, we seek advice from a committee of external experts. And when we do that, all of the meeting materials get posted online. The background material posted for the meeting is usually quite a bit shorter than the full review, so that's convenient. And the backgrounders from both FDA and the drug company are posted ahead of the meeting, and the committee meetings can be viewed live via webcast when the recording is posted online after the meeting. Actually, as a side note, one of these meetings, we call it, they're advisory committee meetings. We're having an advisory committee meeting next week to discuss the clozapine REMS program. So that could be very interesting if anybody wants to listen into that. If the drug in question is one that we don't end up approving, this is also the only public source for information about our take on that application. Because if we don't approve the drug, we send a letter to the company outlining why and what they can do to give us the information that we would need to approve it. But that letter doesn't get made public unless the company chooses to release it themselves. And let's face it, most of the time they do not. But if it's an application that was discussed in an advisory committee meeting, then you still have some way of getting insight into our decision-making process. All right. And although you wouldn't know it from the New Amsterdam storyline, we have been paying attention to clinical trial diversity for at least a few years now. Beginning in 2015, we started publishing snapshots for all approved new molecular entities. So any drug that has not been previously approved in any form. And on this site for the drug trial snapshots, we have a searchable database of all new molecular entities approved since 2015. The snapshot includes a breakdown of the study results by age, race, gender, along with a very brief lay summary of the benefits and risks of the drug. And so this is probably most relevant for folks who are actively engaged in research and conducting clinical trials. I mentioned earlier that FDA publishes guidance documents to help facilitate drug development. So these are all posted online and relatively easy to search. All right. So I also want to take a moment to encourage all of you to submit adverse event reports to us. Our post-marketing pharmacovigilance efforts only work if folks are actually reporting things. So this is particularly important if you observe an adverse event in your patient and it's something that isn't currently labeled. The more data that you can include in that report, the better. And if you do submit an adverse event report to us, don't be surprised if someone from our pharmacovigilance team ends up contacting you at some point for additional information. When they're trying to evaluate a potential safety signal, they often dig into the individual reports and try to follow up to understand the cases better. All right. And finally, I would not be doing my job if I didn't throw in a pitch for new reviewers. So, I really can't imagine having a cooler job. So if you enjoy research, study design, digging into data, etc., basically, if like me, you're a bit of a nerd, you might also think that the job is pretty darn cool. Now, obviously, with changes in administration, sometimes there can be changes in hiring processes and things like that. But in general, if my presentation has piqued your curiosity at all, please, feel free to send me your CV and a short cover letter, and I would be happy to talk to anybody more about who might be interested in a career at FDA. So, thank you. All right, so I think we do have plenty of time for questions. So I don't know, Dr. Rudorfer, did you want to moderate, or should I just go straight into the chat? How do you usually work your Q&A sessions?
You can go into the chat.
Okay. Let's see. Could you comment on the FDA's position on the everyday clinical practice of off-label use? We don't actually have a position on that. I mean, it would be it's off-label for a reason. We're able to regulate the information that goes into the drug label. We're able to write a label based on the clinical studies and say what the FDA approved indication is. Anything beyond that would be considered practice of medicine and is outside of our regulatory authority. So let's see. And then this one that says we also make sure they don't hide failed study data. I'm not sure what that was in. Not sure the context for that one, but Dennis Osden, did you want to? Was there a question in there, or was that just kind of a peanut
Yeah,
gallery?
hi, this is Denise speaking. No, it was just a comment that when you were saying you are making sure about safety and efficacy data submitted by pharmaceutical companies, I just wanted to say that sometimes they may have tendency not to submit everything they have. So we also make sure that they don't hide any failed study data.
Yeah, well, I mean, that's these days, we have clinicaltrials.gov, so they have to register their clinical trials in advance. And we do, we have to give the green light for the trials themselves to say that they're safe to proceed. So we know that they are conducting a study. We've seen the protocol, we've reviewed it, and we'll be looking for that data in the new drug application. They have to submit everything to us. And, one thing that I didn't mention that I probably should have is that as part of the review process, we also have inspectors. We have an entire team of folks who, now granted, we can't inspect every study site. A lot of times you have, 40 or some odd sites for, a large clinical study. But we'll choose, we'll use an algorithm based on, the size of the study site, past history of inspections, whether a particular site is, large enough to have an influence on the overall study results, just, various factors like that. And we will send inspectors to the site and actually review the source data. So not just the database, but also all of the documents that are kept by the investigator, all of the original copies of the clinical outcome assessments, everything, and we confirm that source data and what's in the database actually match. So it's another aspect of our oversight in addition to that. So yeah, so we get all of the data, and we're actually the only regulatory authority. EMA doesn't do this, Health Canada, none of the other regulatory authorities get that raw data. They just rely on the analyses that the drug company conducts. And if they have questions about those analyses, like EMA can stop the clock and ask questions and have, the drug company do additional analyses and submit those. We don't stop the clock. We do the analyses ourselves. And we're the only that does that. So it's very much a trust but verify, with lots of verifying kind of situation.
Thank you. Great.
Okay. I see another Ian Finnegan has a question.
Hey Doc, how's it going? Thanks for a great talk. I have a question about valid controls and placebo controls, and the FDA's opinion on this. So I'm going to use Cobenfy as an example, recently approved drug for schizophrenia. So this drug was approved based on, like a couple randomized trials where the control was they were placebo controlled. So now, the reps are coming out and they're marketing this, and they're telling us, hey, like, after you guys have one, first line antipsychotic fail, you guys can try this. And the issue is that the randomized data that exists for this drug is versus placebo. The standard of care was not involved. So we have data that's saying that it's safe and that's effective compared to placebo, but we have no data on where Cobenfy actually fits into current clinical practice and how it fits compared to, other drugs that, we currently use and are currently effective. Do you see a role for the FDA in pushing these companies to get better data on efficacy compared to current standard of care moving forward? Or do you think it's kind of the current status quo is going to be how it is? Thank you.
Yeah, I mean, the trouble is that we can't actually require superiority studies. We don't have the authority to say that a new drug needs to be better than existing therapy. Depending on if for instance, the company was asking for a claim to treat to treat treatment resistant depression. So for instance, like if you think about esketamine, they wanted that treatment resistant depression indication. And so they enrolled folks who had already failed a couple of antidepressants. But in the case of something where you're just saying, do you or do you not treat schizophrenia, MDD, bipolar disorder, then we really only have the authority to ask for those placebo-controlled studies. At a certain point, if it gets to the point where the standard of care is so well established that a placebo-controlled trial would be considered unethical, then that changes. So, for instance, you would never do a study of a new seizure medication without having, an existing seizure medication on board, right? But yeah, we don't really have the authority otherwise to ask for superiority studies.
Okay, thank you. I think that is kind of the point I'm getting at, though, because I think the FDA currently requires those approval trials to be done with placebo, and I think we are getting to a place where the standard of care is pretty well established. Yeah, so something to think about. Thank you for your time.
Tiffany, I have a question, somewhat related to the placebo issue. Every once in a while, we hear from an advisory committee meeting or otherwise that a trial was done, and while it was placebo controlled, it seemed to be an open secret that the subjects knew whether or not they were receiving the active compound. I mean,
Like
how
one
concern is that?
recent example, for instance, yeah. I mean, it is a challenge, right? When we have these products that have this dramatic functional unblinding, and trying to figure out how we what would be considered an adequate and well-controlled study in that situation, right? So one of the things that we've done, we've actually published we've published a guidance. There's a guidance for that. We've published a guidance about the psychedelics and offered some advice on that. So, for instance, a placebo-controlled study in that context is still important for characterizing safety, right? Because, if you have an active comparator, first of all, even the active comparators, people know if they've gotten a benzo or if they've gotten niacin or whatever, because even if you're trying to mimic some aspects of the psychedelic, there's nothing that really compares to that. So all you're doing with that, active comparator is, an active being kind of questionable. The only thing you're really doing is kind of muddying the waters for your actual safety assessment. But an alternative to that could be to have a dose response study where you have low dose, mid dose, high dose of the psychedelic, in this case, in that single study. And if you see a clear dose response, then that is also supportive of the idea that the drug is in fact doing something. And, you can also evaluate a dose response for the safety signals in that as well. So that kind of the use of those sort of complementary designs so that you can have, you can fill different pieces of the puzzle through different designs. You don't have to answer every question in each trial. You can answer some questions with one and some with another and put together a picture that could ultimately support a new drug application.
Okay.
But yeah, that was a very recent, very high profile example of an advisory committee with that topic. Other question. Oh, does FDA evaluate physical treatments like TMS? We do have the Center for Devices and Radiologic Health. So TMS would fall under the devices review. Now, their standards are, they have an entirely different regulatory authority, and their standards for approval are very different. They don't have the 2-trial standard. They have different classes like class 1, class 2, class 3 medical devices based on, you know, a risk assessment and how that risk, you know, how much evidence they need before marketing depends on how high the risk is. And to be honest, I don't know enough about the the device approval process to even scratch the surface of that. But I would say that it is very different. Even the idea that they have this 510K process where a device just has to be, I think it's substantially similar is maybe the language, to a predicate device. So if there's a device that's out there and approved and you have all of your, you know, engineering data that says this new device is similar, it doesn't even necessarily mean that it has like the same use as the other device. It could be an approved device. It's a very different world than the world that I live in. Yeah, the off-label question is that it's outside of our authority because we don't regulate the practice of medicine. So that was the short version of the answer.
Can I ask a follow-up to the off-label issue? There's been a recent increased interest in treatment of ADHD in adults. And we know there are a couple of meds on the market that have been approved, but only in kids, like guanfacine, for example. I believe the label says approved up to age 17. So what would be involved in expanding that to adults? I mean, would that be like a totally new drug application that a sponsor would have to come in with?
Yeah, I mean, I think we also have a draft guidance for ADHD, as it turns out, but our draft guidance for ADHD is mostly about stimulants. But you know, some of the same principles apply. You know, for ADHD, we always want the pediatric data first because, you know, I mentioned the Pediatric Research Equity Act that allows us to require post-marketing studies for pediatrics, but if it's reasonably likely that that drug is going to be used in a pediatric population, so in ADHD, where it's a disorder that begins in childhood, we do not grant waivers or deferrals for those studies, and so we expect that data up front. Now, if a company establishes substantial evidence using 2 studies in a pediatric population, that's great. They can get their initial approval. At that point, we do think that the disorder is pretty much, it's the same disorder across a lifetime. It may have a slightly different presentation in adulthood, but you know, we think that it's similar enough that a single additional study in adults is all that you would need to expand that indication. You know, so it's an abbreviated program, but it still would require another clinical study.
Okay, thanks. And any other questions? This is your big chance.
Tiffany,
Yes.
What
Hi,
is
Henry.
the most frustrating thing about your, you really told us how fun it is, and you're also recruiting, right? But what is the most difficult part of your job for you and your team?
Oh, I mean, honestly, the most difficult thing is just the volume of work. I mean, it's one of the reasons why I'm always going to put in the plug for new reviewers, because the more that we can spread out that work among more team members, the easier it becomes. I mean, on the one hand, it's when I first started at FDA back in 2010, it was a really kind of a dark time for psychiatric drug development. You weren't seeing a lot of novelty. A lot of companies were getting out of neuroscience altogether. You know, it was, let me see. So it was 2 weeks before I got promoted to my position as deputy, before I got my first new molecular entity, new drug application assigned to me, which, you know, was fun to be, you know, doing my first NME NDA while I was learning a new job. But, you know, that just gives you an idea of how little innovation there was at that time. So now there's a lot more. You know, we've got new molecular entities left and right. We've got new mechanism of action. We've got, I mean, a good third of our workload is psychedelics at this point. There's just so much going on. So it's exciting. And I think that ultimately is going to be great for our patients. I mean, I look at things like, you know, brexanolone and zuranolone for postpartum depression, or, you know, when esketamine first came through the door, I was like, oh my God, this is amazing stuff. I can't believe that we have something that's going to, you know, provide that kind of relief that quickly. There are just things that are so cool in the pipeline, but it's good news for patients. It's good news for public health. It's just really exhausting because there's just so much work to do. I mean, I was telling Kathy before the rest of the crew joined, because I hadn't responded to her email about, you know, is there anything you need at the last minute? It's like I was in meetings from 8:30 till 5:30 nonstop today. Just, you know, it's your tax dollars at work for sure. But I think that's the most frustrating part, is just that there's just so much, and we're trying to do so much work with such limited resources.
Thank you. This was great.
Well, I hope folks enjoyed it, and I hope folks learned a little bit, and, you know, hopefully I told you something you didn't already know. That's usually a good sign. So
Yes, this was terrific. Thank you so much, Tiffany.
Great job. Thank you so much. The CME, the information to claim your CME credit will be forthcoming. So please be on the lookout for that email. And thank you, Dr. Farchione, for such a great presentation.
Thanks, everybody.
Bye.
And next month, our Wednesday evening webinar will take place the first Wednesday of the month, December 4th. So watch out for details.
And it will be a 7 o'clock meeting in lieu of 7:30. So, but we'll be putting out some information for you for that.
So pay attention. Okay.
And
Bye, everybody. Happy Thanksgiving.
Yes, happy.
And Kathy, the recordings will be posted, right? And will be shared.
They will. They absolutely will. Thank you for reminding me I need to stop the recording now.